Dr. Eric Kandel is the leading neurobiologist of our time. He shared the Nobel Prize for Medicine in the year 2000. His pioneering work has helped us understand the brain. It's helped us understand memory. It's helped us understand the relationship between biology and psychology and psychiatry. He is now at Columbia University. I am pleased to have him on this program to talk about this extraordinary journey he has taken from Vienna to Brooklyn, to Harvard, to NYU, to Paris, to Columbia, all the time with a giant curiosity to understand memory and the loss of memory, everything there is to know, and the biological basis of memory. That's been his life's work. That's why he won the Nobel Prize or shared it with some others. And I am pleased to have him here to talk about that on this program. Welcome.
Thank you. I feel very privileged to be here.
Thank you. Let me go back to Vienna, first of all. I mean, you born in Freud's town.
That's right.
You know, 10 years later you leave.
That's right.
1939, fleeing the Nazis. How'd you get out?
Well, it's remarkable. My parents were fairly simple people. They ran together, ran a toy store in Vienna. But my mother was very prescient and sensed that as the discussions were going on about a possible plebiscite, Schuschnigg, the chancellor of Austria, sensing that Hitler was putting a lot of pressure on him, declared a plebiscite to determine whether or not Austria should remain free or join Germany in a pan-German speaking union. Hitler realized that Schuschnigg was probably going to win, so he marched in. But in those several days in which the plebiscite was actively being discussed and people were preparing to vote, my mother sensed that it might not come out well. And she went to the American embassy and inquired how one leaves, how one goes to America. She had a brother in the United States, my Uncle Berman. And she was told that one needed to get an affidavit. Somebody had to guarantee your support once you came to the United States. So she wrote to her brother and got from him an affidavit, and by the time that arrived, Hitler had already taken over Austria. A quota was established. There was an American quota, which was now very rapidly filling up, and since my parents came from Poland, it took a year before our number came up. And even as it came up, we had to leave in stages. So my grandparents left first. They left in February. My brother Louis and I left in April, and my parents didn't leave until September. My parents left a few days before the war broke out.
Reunited in Brooklyn?
In Brooklyn, yes.
Then Harvard College?
Later at Harvard College,
Yeah, and then went on to Harvard College later?
Yes. When I first came here, I went to elementary school. I first went to PS 217, and then with some tutelage from my grandfather in Hebrew, I transferred to a very good Hebrew school, a Jewish parochial school called the Yeshiva Flatbush. And I went there for 4 years until I graduated, went to high school at Erasmus, and from Erasmus I went to Harvard College.
Yeah. Some great athletes came out of Erasmus High School.
That's right. Sid Luckman came from
Yes, exactly right. And others. Some great basketball players, too. Why Harvard, and then why NYU?
It's really extraordinarily interesting. So I thought I would go to Brooklyn College from Erasmus, which is where most of the academically oriented kids went to, and I had applied to Brooklyn College, and John Campana, one of my history teachers, approached me and said, have you ever thought of applying to Harvard? And I said, not seriously. He said, well, why don't you think about it? So I discussed it with my parents, and they thought, gee, you know, you're almost certainly going to get accepted at Brooklyn College. It would cost additional funds to apply to Harvard, to fill out the forms, and they discouraged me from doing it. So I told Mr. Campana that my parents didn't think this was a good idea, so he put out the money for me to apply to Harvard.
Wow.
And Ronald Berman and I were the two people from Erasmus, the class of 1400, accepted at Harvard and on a scholarship. I mean, that's fair Harvard indeed.
Yeah. And studied history? Or?
I majored in an area called history and literature.
Yeah.
And I was very much interested in European intellectual history. I was interested in the kinds of issues that had concerned me. And I wrote my dissertation on the attitude toward National Socialism of 3 German writers, Carossa, Zuckmeier, and Junger, fairly obscure people to the American public. But they were interesting because they had different positions vis-a-vis the Nazis. So Zuckmayer was a very progressive dramatist who actually was in Vienna. He was a German dramatist who had written some satirical dramas about Hitler, escaped from Germany, went to Austria, and then when Austria was invaded, he came to the United States. Carossa was a physician who took an interesting position, what he called a inner immigration. That is, he stayed in Germany, but philosophically he was opposed to the regime. And Jünger was really a proto-Nazi. He had been in the First World War. He loved the idea of battle. He thought this brought out the best in men. And he was really one of the intellectual precursors for the Nazis, although he later had some disdain for their vulgarity. So I sort of contrasted their different political positions. And one of the things that sort of struck me was how weak the intellectuals in Germany, and certainly in Austria, proved to be in the face of Hitler. They didn't provide very much opposition at all. A number of people, including my colleague at Columbia, Fritz Stern, feel very strongly that if there had been more opposition to Hitler in the first few months when he really didn't have complete control of the government, his position could have been weakened a great deal.
Yeah, there's a lot of criticism to go around in terms of when you look back, in terms of what might have happened and who might have stood up. And it's easier to make those points. Today, you, coming full circle, still look back at memory and painful memory and the experience of memory to try to understand how there is a chemical or a reaction within that process.
Yes, yes. I mean, it's hard to say exactly what motivates one's intellectual life.
Right.
But certainly my experiences with Hitler in that 1 year, 38 to 39, made a dramatic impact on me. I was really frightened through much of that period. All my friends who were not Jewish stopped talking to me the day after Hitler came in. I was ostracized in school. I was roughed up in the park. My father was arrested for several days, and we didn't know where he was. During Kristallnacht, that was the night in which all the synagogues in German-speaking countries, in Germany and Austria, were destroyed and burnt. We were kicked out of our apartment, and we were asked to live with another family. And when we returned to our apartment, everything was gone. Everything of value was gone. So that year was a very difficult year for me. Later on in college, I tried to sort of understand that from a historical, intellectual point of view. And in part through some acquaintances that I made at Harvard, I got interested in psychoanalysis. And
as an undergraduate?
As an undergraduate at Harvard. And you're probably too young to appreciate this, but in the 1950s, psychoanalysis was the way to understand the mind. Yeah. It was really an exciting new intellectual development, and it contrasted to behaviorist psychology that was really interested in very simple aspects of behavior and had a sort of mechanical view of how behavior was generated. Analysis had a much more sophisticated, nuanced understanding of mind. And I decided that if I wanted to become an analyst, I would go to medical school.
But the point is, you wanted to become a psychiatrist, a psychoanalyst.
Absolutely.
That was where your interest was.
That was my interest. That was absolutely my interest. I thought to do psychoanalysis would be the most fascinating way to spend one's life.
And you were a huge admirer of Sigmund Freud.
Absolutely. Yeah.
And today?
I still admire Sigmund Freud. I think he's a genius.
Because even though the shift has been away from psychoanalysis to a molecular biology basis.
Freud discovered the existence of unconscious mental processes. I mean, this is really one of the great insights into mental life. So much of our life is unconscious. He had insights into the nature of conflict and what determines people's actions. He felt that everything about people's actions had a specific meaning that may be manifest or may be latent. I mean, this richness of understanding is extraordinarily worthwhile. The issue is not the value of those insights. The issue is, can we test them rigorously in some independent method? And I think that's the issue in psychoanalysis today.
And that's the issue of your own scientific exploration?
In a very modest way, yes.
In a modest way?
Yes.
Why do you say modest?
Well, I don't address the issues of the human mind directly. I do them through very reductionist approaches using very simple animals.
snails.
So when I look at a snail, I don't pretend to know what's conscious or what's unconscious.
We're going to come to the snail in just a moment. All right, so you there get ready to go to medical school because you want to be a psychiatrist. You go to NYU.
That's right.
You know, and when did you get interested in this study of not becoming a psychiatrist in the tradition of Sigmund Freud, but also becoming a researcher in the tradition of...
You know, in retrospect, it seems like it's sort of a predetermined trajectory. It was really much more accidental than it seems. I felt that even someone interested in psychoanalysis should know something about the brain.
Right.
And in NYU, while I was a medical student, we were given the opportunity to take an elective period. And I took an elective not at NYU, which didn't have any neuroscience to speak of when I was a medical student. I went to Columbia, where they had a superb neuroscience group directed by Harry Grunfest.
Right.
And Grunfest had me work with one of the young associates in his group, a guy by the name of Dominic Purpura. And he was exploring how certain modulatory systems in the brain worked. And I found that just fascinating. And I remember having dinner with Denise, whom I later married,
Yes.
a French woman who had come to the United States a few years before that, and saying to her, you know, this is an amazingly enjoyable way to make a living. But, you know, it's ridiculous for me to think about this seriously. You and I don't have any resources. It's going to be important for me to have a private practice. And she said, that's ridiculous. She said, my father always wanted me to marry a poor intellectual. I think we should do exactly what we want. That was just absolute. She has not said that since.
You have been blessed by two important women in your life, your wife and your mother. Your mother got you out of Vienna, and your wife says you got to stay in research.
Absolutely. In fact, you actually have put your finger right on the critical issue. Both those women have been extremely important. And in the later part of my life, Denise has had an absolutely critical role.
When did you figure out that the best way for you to study the science of the brain was to study the snail?
That really was several years after I first made the decision to look seriously to becoming
And then I want to figure out what the snail has been telling you.
Okay. So we're now talking about 1955, 1956, as I was finishing medical school. I enjoyed that experience a great deal, and as a result of that, Harry Grunfest asked me whether I would be interested in having him nominate me for the National Institute of Health. The National Institute of Health had a direct connection with the Public Health Service. It was part of the Public Health Service. And so physicians in 1956-57 were being drafted for the Korean War. They had an option if they were interested in research, capable of doing research. If they were selected by the NIH, there was alternative duty. And so he nominated me to the Laboratory of Neurophysiology at the NIH, and Wade Marshall, the director of the laboratory, selected me exclusively in Harry Grunfest's recommendation. So after a year of internship, Denise and I had already gotten married. We moved to Bethesda, Maryland, and I worked at the NIH for 3 years. I was originally going to stay for only 2, but ended up staying for 3 years. And that was a transforming experience, the most important scientific experience of my life.
Because?
Being at the NIH is like being in a biological nirvana. It always has been, but in that period, that was really the golden age of the NIH. Young people, relatively naive physicians, came there and were transformed into functioning scientists. It was really a remarkable opportunity. And I learned how to do science. I learned how to think about scientific problems. I learned to try to develop ideas of my own. And I began to focus in on memory as a central problem in biology, and particularly, I thought, the central problem of psychiatry and psychoanalysis.
Because?
What one tries to do in psychoanalysis to understand how previous memories affect your current behavior.
Right.
And to get some insight into how memory is stored seemed to be a fantastically interesting problem. These are issues that previously were in the realm of philosophy. And then in the realm of psychology, we had very little biological insight into what are the detailed mechanisms whereby memories are stored in the brain. And just as I had arrived at the NIH, so this was 1957, a remarkable paper appeared in the scientific literature on a patient called H.M., who had been studied by two people, a neurosurgeon by the name of Scoville and an outstanding psychologist, Brenda Milner, a friend of mine, just extraordinary contributor to science. H.M. had been hit by somebody riding a bicycle when he was 9 years old, knocked unconscious, sustained a brain injury on both sides of his brain, in temporal lobe, that gave him seizures. At first, the seizures only interfered to a minor degree with his functioning, but as he went through elementary school and then high school, it interfered more and more. And ultimately, he presented himself to Scoville, who was a neurosurgeon, for surgical removal of the tissue that was causing the seizure. That surgical removal essentially cured or substantially improved his epilepsy, but it left him with a profound memory loss. And that memory loss was so interesting that it drew everybody's attention to it. And one of the regions that was removed from that patient's brain as a result of the surgery is a brain region called the hippocampus.
All right. Right. And so you started to study the hippocampus.
I started the hippocampus because of that.
And is that how you discovered, everybody discovered, that memory was stored in the hippocampus?
That was the first insight, that the hippocampus had a role in memory, and it also defined two additional things. It defined the specific nature of the hippocampus in memory storage. It had a very interesting role. So it doesn't have any relationship with short-term memory, the memory that one uses when one learns name Charlie Rose. It doesn't interfere with memories that have been stored for a long period of time. H.M. remembered his childhood, later years perfectly well. It interferes with the acquisition, with the transfer of new short-term memory into new long-term memory. So it not only gave a function for the hippocampus in memory storage, but it showed it had a storage capability that was important for taking new information and transferring it into long-term memory. And it showed that a lesion of the brain could leave everything else intact except the transfer of short-term to long-term memory. So H.M. had cognitive capabilities. He could recognize things. His IQ was not changed. He could read. He could write. The only defect he had was a defect in memory. Lastly, a number of other people thought that memory was tied up with other processes, could not be studied in isolation. So it not only defined a specific function for the hippocampus, but made one realize that memory was a unitary function of the mind, that it had a specific role. It could be separated out from others.
Let me, I was going to ask you this later, but I'll ask you now. You once said, if you can describe, if I could describe what is happening in your brain as you look at a picture of Mona Lisa, then the problem of memory is almost solved, the issues of memory. What did you mean by that?
That is, I was posing an enormously difficult task for biological sciences. We are very far from understanding how we appreciate complex visual objects, how we appreciate great works of art. That if we understood the brain well enough to understand how we picture in our mind's eye a great work of art and how we remember that, we would have really many of the mental problems that confront us. We would understand them in appropriate cellular and molecular terms.
Having asked that, let me go to the snail.
Yes.
Where you had to go to Paris and you worked with a great expert. What was the name of the snail?
Aplysia.
Aplysia, okay. Right. A small, simple molecular structure or not?
Right. So the reason I went to Aplysia is because I was interested in studying the brain from the point of view of how individual nerve cells behave.
Isolate and locate and focus on?
To work out a simple reflex that could be modified by learning, so I could see how memory is stored in that simple reflex organ.
And so you would go tap the tail of the snail?
That's right.
And what would you find out from that?
And I would find that if you tapped that repeatedly, the animal would ignore the stimulus. It would become weak and weaker.
So in the beginning, if you tap it, it would move, and then after you tap it,
that's right.
It would stop moving.
But if you now gave it a noxious stimulus and scared it, that same weak tap would cause a powerful response. the animal would become frightened and it would give you an enhanced reflex response. So we had two different forms of learning. One, a decrease in reflex strength, and the other is a tremendous enhancement of reflex strength. And we could see what was the underlying change that occurred in the brain.
So if you find the right animal, you can learn that is simple enough to comprehend because there are not so many cells at work. Help me understand this.
That's exactly right. There were two great advantages to aplasia. One is that there were very few nerve cells, so you could trace out the neural circuitry in terms of individual nerve cells. And two, for reasons no one understands, many of these marine snails, particularly aplasia, have gigantic nerve cells. So you could see them with your naked eye, and you could see the same cell repeatedly in every animal of the species. That really made it much, much simpler to study the problem. And that is the strategy that biologists have used since the beginning.
This is part of understanding the process of scientific discovery, too.
That's right. Reductionism has been the key to scientific advance in every area of biology. Somehow, when it came to mental processes, biologists, not so much the biological community, the psychological community was initially reluctant to think this would be an effective approach. But it turns out that there is such conservation in evolution that if you get deep insight into any process in any species, it's likely to be of general importance.
Okay, this lifelong collaboration with this little snail has continued for you.
Yes.
He's still your friend.
Yes, very much
or
so.
She's still your friend, or whatever.
Oh, it's everything in my professional life. That is.
So did you mention the snail in your Nobel acceptance speech?
I showed a picture of the snail with the Nobel Prize around the neck. That snail deserves the Nobel Prize.
That's great.
That's a wonderful
All right,
snail.
tell me in the 5 things, just take 5, the most important 5 things you've learned from this simple snail about memory.
Let's take one form of learning, and that is sensitization. The enhancement of reflex response by giving an aversive, offensive, noxious stimulus to a part of the animal's body. We found that when you produce sensitization, when you frighten the animal and you produce enhanced reflex response in preparation for defense and escape, you strengthen the connections between certain cells that make up the neural circuit of the behavior. The behavior involves sensory neurons that pick up from the skin and motor neurons that move a particular organ called the gill. The connections between the sensory neurons and the motor neurons are fixed. Every animal has that connection between the sensory neuron and the motor neuron. What is not specified is how strong that connection is, how effectively the sensory neuron is in firing the motor neuron. And what learning does is to alter the strength of that synaptic connection. When you alter it for a short period of time with short-term memory, you produce a transient strengthening of that synaptic connection that lasts for minutes, and it wears off as the memory wears off. When you produce long-term sensitization by repeated training, just like you and I learn by repetition, practice makes perfect. So the snail learns by repetition. If you give this offensive stimulus repeatedly, the animal will remember that offensive event for days. That involves a fascinating long-term change in that synaptic connection. And what is fascinating about that are 3 features. One is that involves an alteration in the expression of genes in the sensory cells. So when I talk to you and we have this conversation, insofar as you remember anything about this, and I'm not urging this on you, I'm just describing a possible
it's not
outcome.
Required for our friendship.
A possible outcome of this conversation. It is because genes are being altered in your brain.
Genes.
Genes in these particular nerve cells. This will not be passed on to your progeny because this does not occur in the sperm, it does not occur in the egg. This occurs in certain neurons of your brain that are responsive to the stimulus. Why do you turn on genes? The reason you turn on genes is because ultimately the way the connection is strengthened and maintained strong is because you grow new synaptic connections. So instead of having, let's say, 100 synaptic connections from sensory neuron to motor neuron, after long-term sensitization training, you have 200 synaptic connections. You have a doubling of the number of synaptic connections. Now, some of my friends worry about that. They say, my God, my brain is going to be just stuffed with these new connections. And I tell them, not to worry.
Not to worry.
Not to worry. First of all, we forget,
Yeah.
and the connections regress.
They do.
And
They regress.
Craig Bailey at Columbia showed. You
Okay,
see
now what about between smart and not so smart people? A difference?
That's a different part. We don't know the answer to that. We don't know whether or not this is enrichedness of connections.
Right.
There could be other possibilities. For example, we found that in switching on these genes, there are inhibitory constraints on memory. There are molecules in cells that work to prevent the memory from being turned on.
Yeah.
So what you do when you turn a memory on, when you throw this genetic switch, is you not only have to activate certain genes, but you have to remove these inhibitory constraints. So one way you could get people to be super smart is if they don't have a very effectively working inhibitory component of the system.
All right, now let me get to this too, because where we're going here, I hope, is that in working and understanding these chemical reactions and what happens
Yes.
to these
Yes.
connections, we may very well find later a drug or a chemical that can influence the retaining of memory, which, as we know, I think, decreases as we get older.
Absolutely. But before we get to that, let me return to
Okay.
psychoanalysis
All
for
right.
a second, okay? How could we make psychoanalysis a hard science, a science that you and I would be intellectually satisfied with?
Right.
Brain imaging is coming of age. We now can image the human brain, we can image it in action. So let's say we image my brain.
Right.
And we find that
We've
my,
all seen these pictures and showing
Right,
what's going
right.
on in the
and
brain.
we find that my superego is too large. This is not true, but we can then decide if I, am a good patient for psychoanalysis. And imaging techniques, and I'm now
Right.
speaking about the future, imaging techniques have achieved the degree of resolution where people could figure out where these different structures are located. And superego is not going to be at one site, it's going to be a bunch of different sites. We could determine that there is an abnormality here in superego structure or whatever.
Right.
And we could see whether or not the psychoanalytic experience really has the anatomical effect that we would expect to bring a shrinkage of the superego. Now, I'm giving you an extreme example. But we find in humans that as they learn tasks, they undergo anatomical changes in their brain. This is something that Mike Mersnick first pointed out for
Right,
higher
right,
animals. He showed that if you look at the representation of the hand area, everyone has a representation in the brain of the whole body surface. If you look at the hand area of different monkeys, each monkey has a somewhat different hand representation. And he didn't know, is this due to the genes or is this due to experience? And so what he had the monkeys do is to press a ball for food with 3 fingers of their hand. And he found after several weeks that the representation of these 3 fingers was expanded at the expense of other areas of the brain.
All right, you got to go through that one more time for me.
That if you use certain parts of your body,
Right.
the representation in the brain of those body parts will be enhanced. If you look at the representation of the left hand of string instrument players, you find that their representation is richer than those of people who don't play a string instrument because they individuate their fingers and they use them independently. The right hand, which just bows,
Not the same.
doesn't have that. And people who start playing string instruments before puberty, before age 13, have a larger representation than those who start playing later on. That's very profound, Charlie, because that means that this could contribute importantly to the biological basis of individuality. 2 identical twins having exactly the same genome will undoubtedly have somewhat different life experiences. And those different life experiences will be reflected in the structure of their brain. So every individual that you encounter has a slightly different brain than any other individual.
Because
that you
their
encounter.
experiences are different in the representation.
That's right. So it's a combination of genes and experience. But even identical genes, people will have slightly different brains.
Okay, now I may be getting ahead of myself here, but you injected some mice to enhance memory, correct?
Yes.
With something called roloprem.
Yes.
And what was the result of that?
They were able to improve their memory. Yes. So let me go into that.
Okay.
Okay.
And is that, should we talk about this here, or is this the right time to talk about that?
Yes.
Okay.
Yes. Yes. And maybe I can
make the
provide
connection.
a little bit of an intellectual transition. So in Aplysia, together with a substantial number of colleagues over the years, we were able to get some initial insight into the cellular and molecular mechanisms underlying these very elementary forms of memory storage. And obviously it became of interest to see to what degree one can now extend this kind of methodology to more complex forms of memory. I had been working only with the kinds of memory that HM had, what is called procedural, reflexive, or implicit memory storage. The conscious memory, which HM lost, you cannot study in the snail. But you can study it in the mouse because the mouse has a hippocampus.
Right.
In the 1990s, two scientists, Capecchi and Smithies, developed techniques for knocking out genes in mice. It was just a major revolution. One of the many.
Knocking out genes in mice. What does that mean, knock them out?
You can eliminate at will any single gene, its function.
Right.
And it became clear to me, as it did to many other people, Susumu Tanagawa, a number of other people, that this is going to be wonderful for looking at memory storage in the mouse. This opens up a molecular exploration of the mouse, because not only can you knock out genes, you can express individual genes in the mouse. So my colleagues and I began to explore hippocampal-based explicit memory, the kinds of memory that in humans involves conscious recruitment for recall, conscious participation for recall. And we found that if you look at the hippocampus, you see that synapses change within the hippocampus. One had known this from earlier work, not done by me. And we began to look at the molecules involved in those changes, and they turned out to be amazingly conserved to those that we had encountered in Aplysia. Now, one of the signaling systems we had encountered in Aplysia was a system called the cyclic AMP system. And roloprem is a drug that elevates the level of cyclic AMP in nerve cells. And the reason we were interested in using roloprem and other drugs that increase the level of cyclic AMP was that we had found, as had others, that mice as they age undergo an age-related memory loss.
Right.
Very much like humans. You know, some humans, unfortunately, go on to develop Alzheimer's disease, but many people as they age undergo a slight weakening of memory with age. It's called benign senescent forgetfulness. For people who suffer from it, it's not so benign, and it doesn't necessarily begin in senescence. It begins in mid-40s. Right. And with mice, you find exactly the same thing. Mice live about 2 years, 12 months of age. They begin to show a decline in their ability to perform certain hippocampal-based spatial memory tasks.
Can't work their way through the maze.
That's right. And Mark Berard in my lab and Elizabeth Bach found that if you increase the level of cyclic AMP, either by giving roloprem or by giving other substances that increase the level of cyclic AMP, you can restore the physiological deficit and, to a large degree, the behavioral deficit in these mice. And with the help of Columbia, I've recently participated with Wally Gilbert and Jonathan Fleming in starting a company called Memory Pharmaceuticals that is designed to try to take advantage of these early findings to develop drugs.
To develop drugs?
To develop drugs.
Do we have any drugs now?
No.
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 And how far off is that? So that you can take a drug that will enhance your memory and reduce the onset of loss of memory, or the progress of loss of memory. How far away is that?
You want an optimistic?
Yeah, give me an optimistic. Give me a range.
I think we are not.
We're talking about 3 to 5 years?
I think within 5 years there's a good chance we will have a beginning therapeutics in this area. That's a hopeful estimate.
So that we will be able to take drugs suppose you've had certain memory loss. Will you be able to regain
I'm not sure
memory?
of that, but you will be able to acquire memories more effectively if you have difficulty with it and to retain it better. Whether it will help you recall information that you've already lost is not at all clear.
Someone once said to me, and I think it was Danny Hillis, but somebody like that, said to me, understanding the brain is easy. Understanding the mind is difficult. Is that simply because we don't know what the mind means?
No. The mind is a series of functions carried out by the brain. We know quite well what we mean by the mind. Before we understood the biology of the brain, people dealt with mental processes as if they were off by themselves, unrelated to the body. The classical dualist view came out of the idea that there's a biology, there's a body, and there's a mind. Somehow they're not connected. We now know that every mental process has a representation in the brain, not necessarily in a single area, but some combination of areas. But to understand how complex problems are represented in the brain, how complex functions, is an enormously difficult task. Let me give you the example of memory. We have some beginning insights into storage mechanisms.
Right.
But
memory storage, how we remember things.
Right. At specific loci in the brain. But if I want to tell you how I remember the Mona Lisa, that's infinitely more complicated. That means the whole process of visual perception, how does
the image go from through your eyes into your brain?
Into the lateral geniculate nucleus, into the visual cortices, and there are a whole bunch of...
We're nowhere in understanding that.
We're very far away from doing that. I wouldn't say nowhere.
Okay, but
Hubel Wiesel, wonderful tradition of neuroscience that has given us an opening to the problem, but we're nowhere to understanding it completely. But one has to put the cards on the table. When we're dealing with the biology of mental processes, we're not only dealing with the most difficult problem that biology has ever confronted, and biology has now overcome some really fantastic hurdles. The nature of the gene, the development, oncogenesis. I mean, we really have very good understanding.
Okay, but I mean, everybody that I talk to, not everybody, but lots of people I talk to, not just those whose life's work it is, but people like, I remember I recently this summer had a conversation with Larry Summers, soon to be the president of Harvard, said to me, we were talking about what things are happening in science today that will most affect the future, science. He said a number of things. One was the brain.
This is why he was selected to be president of Harvard. Anybody who has judgment like that deserves
to be president.
I tell you, he plays tennis in Wellfleet in the court next to mine.
Yeah, he's a very good tennis player. I know, I've played it. I know, yes, yeah, I know. But my point here is that we're all talking about what is one of the great frontiers of science.
It is
understanding how the brain works, and how, therefore, we can figure out its relationship to pioneering work done, in your case, understanding its relationship to psychoanalysis and memory loss, and how memories are painful, and what we... Help me out here.
I think most people who think about the nature of knowledge consider the biology of the brain, the biology of the mind, the great problem for the next 100 years at least. And young people feel that the attention on the brain will compare to the attention that we gave to the genes.
Oh, that's what I was just saying. So mapping of the brain will be every bit as significant as mapping of the genome.
Yes. Yes.
Don't.
I think it's a more difficult problem, a much, much more difficult problem. The mapping of the genome, which I'm not a specialist, by the time came to it, the task was really a technological one. Here,
in other words, using computers to make it happen fast.
Right. Here, we don't conceptually have a sufficiently good understanding to appreciate the nature of the problem of perception of motor control. So it's a much richer and more complicated problem than mapping the genome.
Yeah, but are all those psychiatrists who went and got these, not only a medical degree, and then went on and had years of residence in psychiatry, are they all saying, man, I wasted my time? Because here comes people like Eric Kandel, and they are establishing the biological basis of behavior. So what am I doing over here trying to understand how behavior comes from retracting and retracing memory, when it's all about chemistry and cellular interaction.
I would make 2 points. First of all, my own perspective on this, you have to realize that mine is a modest contribution. You're speaking to me as a representative of a whole discipline.
I know.
Okay?
Well, you're the only one I got here.
Okay.
You're the only Nobel laureate I haven't round, you know?
That's issue number one. Issue number two, I think those of us who work in the biology of mind don't see this as a way of replacing psychiatry or psychoanalysis.
Let me just repeat my question one more time for you.
Yes.
If I'm a psychiatrist, I may say, here comes Kandel, who's trying to help me by understanding, and all of a sudden, he not only didn't help me, what he has done is made me irrelevant.
I don't think that's going to happen.
Okay.
I see this as a merger of really several intellectual streams. That is of the psychological and psychiatric study of mind, and that of the biological study of mind. Psychology, of which psychiatry is a part, psychoanalytic thinking, psychology, is the framework which we want to explain. These are the phenomena. Memory was defined, short-term memory, long-term memory, were defined in psychological terms before we knew the biology of it. So they give us...
Were they right?
In many of their descriptions, they were. They knew about
Freud
short-term...
was right on many things except women's sexuality.
Yes, he was wrong on that. He was wrong on lots of things.
Right.
But the overall view of the complexity of the mind, the existence of conflict, the existence of unconscious processes, of mental determinism, these are overriding notions that are right. But the point is not whether or not Freud is right on a particular point or not. The point is that psychological thinking, which is evolving, and biological thinking, which is evolving, ultimately want to merge to develop a biologically based psychology that is satisfying from both the psychological and the biological.
A biologically based psychology.
Absolutely right. Absolutely right.
Now,
in all fairness, when people have problems, they go to psychiatrists, and intelligent psychiatrists, well-trained psychiatrists, use their biological insights as well as their psychological insights in order to treat patients. They use drugs, they use insight into interpersonal relationships and combine them effectively in order to treat people.
Are drugs going to be the key to, are we looking at a future in which drugs who will impact the relationship of cells are going to just change everything about the way we endure as human species?
I think biology will give us profound insights into the nature of mind. Those will be accessible to therapeutic manipulations by drugs in much more effective ways than we have right now. But there's no reason to believe that insights into the biology of mind won't give us better pedagogical practices that we have right now, give us better psychotherapeutic practices that we have right now. I do not believe we're heading toward a drug-addicted society. I don't think that's the ultimate goal.
Well, don't we? Okay, but I'm playing this at the layman's level.
Right, but you don't want to replace meaningful personal relationships with a set of drugs. I mean, there is satisfaction one gets out of life, enormous satisfaction that comes out of things that are completely...
Yeah, but I mean, think about gene therapy. Think about all the things. I mean, the horizon, and I'm trying to do this...
We're speaking about disease states. So we're speaking about somebody who has, Huntington's disease or cystic fibrosis. Yeah. We're thinking of gene therapy in terms of people that have defective genes. You and I, as far as we know, don't have seriously defective genes.
No, as far as we know. Well, maybe. I'm not sure about me, but but we are talking about, I'm at the first person at the counter if you tell me there's a chemical that I can take to enhance my memory.
I'm not sure, Charlie, that your memory needs to be enhanced or can be enhanced. What I feel confident...
Can I introduce you, my friend, the rats who've been taking all of this?
They have an age-related memory loss. I'm not sure.
Age-related? I mean, at half of their lifespan, they take it.
I think that elderly people will probably have drugs that will help them. But if you ask me, is this a desirable thing for a young person to take in cramming for an exam, I am very skeptical about that. I think that with every drug, there are going to be side effects.
I know I'm taking... Yeah, okay. And we don't even know that. We haven't begun to understand that. Okay. Any side effects on the rats?
Not in the doses that we're using now, but, we don't... we have very limited...
It's a long way from rats to human beings. I understand.
That's right.
Okay. I know this is far afield from where your primary interest is, but when you think, what about understanding Alzheimer's and what, because that is of the brain, right?
Yes. Well, I think the progress in Alzheimer's disease has been brilliant. I mean, you know, this was a disease we really didn't diagnose until 20 years ago because people didn't live long enough to realize about that. So many misdiagnoses were carried out. We now have a great deal of insight into the nature of beta amyloid, its deposition in the brain, about individual genes that are defective that lead to an early onset of Alzheimer's disease, the progress in the biology of Alzheimer's disease, and on predisposing factors like ApoE that predispose to getting Alzheimer's disease has been brilliant. So if we make progress in other areas of brain pathology, the way we've made progress in Alzheimer's disease, I think we have right to be proud.
Memory is your thing, correct?
Yes.
Okay. We haven't, if we haven't established that, we've done nothing in these 49 minutes. What do you know about photographic memory?
I actually have a slight interest in photographic memory, and it means different things to different
That's
people.
what I want to know. Because I've always been envious of those people who say they have photographic memory.
You should not be envious.
All right.
I used to be envious until I read more about them.
All right.
Let me tell you a little bit about photographic memory. A reason I became interested in photographic memory is because work that Duesenbarge and I did, and parallel work that was done in The Flies by Tim Tully and Jerry Yin, showed these suppressors of memory. That there are certain genes that work in order to increase the threshold, to make it more difficult to put things into long-term memory. Now, why would you want to have a high threshold for putting things into long-term memory? Well, one of the reasons is you don't want to remember everything.
Exactly.
There are lots of things you want to
Exactly.
forget. Okay?
Right.
So we both found that if you remove this repressor,
Yeah.
you put things into long-term memory, boom, like that.
Yeah.
You have flashbulb memories. You have photographic memories. So I began to read up about photographic memories. And there are a number of them described. There's a very famous patient described by, not a subject, not a patient, described by the Russian psychologist Luria, who described this guy who remembered absolutely everything, conversations, rooms he had been in, all kinds of things that happened in his life. This person was quite shallow. Shallow. If you speak to people who have these absolutely fantastic memories, they say, you know, it's like having your head filled with garbage. And the story I most like is the few documented cases of the Talmudists. You know the Talmudists?
Yes.
There are well-documented cases of Talmudists who've memorized every single word of every single page of all 12 volumes of the Babylonian Talmud, only the Babylonian Talmud. They can do the needle trick.
Yes.
You can put a needle in one of these volumes through any single word on a given page, show him where that word is. He counts down, he counts across. You can push the needle out on another page. You tell him what page it is. In his mind's eye, he counts down, he counts across, and he tells you what that word is. Wow. His memory is that remarkable.
That's amazing.
But is he happy?
I don't know.
He is not. He is not a true Talmudist in the sense he does not enjoy the richness of the... He can't play tennis?
Can't play tennis?
So there's a U-shaped function. That is, if your memory is too good, you can't really utilize the information that you have in a really
still,
creative way.
You haven't answered my question. Is why do some people have that and some don't? I mean, let's assume it's not necessarily as good an idea as I thought it was, but why does...
I would
Why can somebody remember more than I can chemically?
Based on what we know right now, that one way you can get that, there are probably a whole bunch of different ways, one way you can get that is to have few of these memory suppressor genes, to have few of these genes that prevent you from putting things into long-term memory.
Now, are we at the point where we can take these memory repressor genes out?
In the mouse and in the fly, yes.
Okay, how about in the human?
Far away.
How far? You don't know.
I don't know.
What do you think? Guess. Just guess.
Well,
not science, guess.
This would not be my first priority, doing gene manipulation with the human
fair
brain.
Enough. Mine either. Okay, but let me come back to mapping. Go ahead, mapping the brain, because that's exactly where the science is about, right?
Yes.
And what we mean by mapping the brain, what?
To understand how different mental functions are represented in different brain regions and combinations of brain regions. Okay?
How many brain regions are there? I mean,
many more than we realize. So Brodmann characterized about 52 brain regions. There are many, many more regions. There must be, in the cortex, the outer layer,
right,
there must be hundreds.
We've always known right-left brain stuff.
Yes.
Right. We've always known that.
But there are many, many more subtle subdivisions.
Okay, I'm way off into locker room.
Well, let me just give you an example to be more specific. In the visual system, right?
Right.
We find out that you process vision by dissecting it into component parts. So there's a part of the visual system that analyzes the shape of your head. There's a part of the visual system that separately analyzes aspect of the coloration of your eyes, your hair, your skin, your suit. There are a separate part of the visual system that analyzes the movement, the tilting of your head. These things are processed by somewhat separate regions and then combined in what is called the binding problem, where these things are brought together in a way we still don't understand to give me the image of Charlie Rose. So there are many subregions for each function that needs to somehow be combined to give us the percept or the action. And this is what the task for the current generation of neurobiologists is.
What do we know about? Go ahead.
And then, of course, there's the deep, deep problem. Consciousness. What are we even talking about when we speak about the nature of consciousness? Now, people like John Searle and Jerry Edelman are beginning, Tony Damasio, are beginning to develop sort of scheme of thinking about this. But the biological underpinning of this...
What does consciousness mean?
It is an awareness, how I focus on you to the exclusion of everything else around you. So selective attention, my attending to you, is the simplest example of consciousness.
I thank you for coming. It's a pleasure to have you here.
Pleasure for me to be here.
Dr. Eric Kandel, Nobel laureate, received the Prize of Physiology of Medicine in year 2000, which he shared with Arvid Carlson of the University of Göteborg, Sweden, and Paul Greengard of the Rockefeller University here in New York for their contribution to neuroscience. He is a university professor of physiology, biochemistry, and molecular biophysics at Columbia. He's also involved with the Howard Hughes
Medical Institute. He's been my generous supporter for a marvelous organization.
Member of you were early on selected to the National Academy of Science. Has won lots of prizes. This has been one of the great hours for me, and I thank you very much for coming.
Thank you very much.
Thank you for joining us. We'll see you next time.
That's right.
You know, 10 years later you leave.
That's right.
1939, fleeing the Nazis. How'd you get out?
Well, it's remarkable. My parents were fairly simple people. They ran together, ran a toy store in Vienna. But my mother was very prescient and sensed that as the discussions were going on about a possible plebiscite, Schuschnigg, the chancellor of Austria, sensing that Hitler was putting a lot of pressure on him, declared a plebiscite to determine whether or not Austria should remain free or join Germany in a pan-German speaking union. Hitler realized that Schuschnigg was probably going to win, so he marched in. But in those several days in which the plebiscite was actively being discussed and people were preparing to vote, my mother sensed that it might not come out well. And she went to the American embassy and inquired how one leaves, how one goes to America. She had a brother in the United States, my Uncle Berman.
Dr. Eric Kandel is the leading neurobiologist of our time. He shared the Nobel Prize for Medicine in the year 2000. His pioneering work has helped us understand the brain. It's helped us understand memory. It's helped us understand the relationship between biology and psychology and psychiatry. He is now at Columbia University. I am pleased to have him on this program to talk about this extraordinary journey he has taken from Vienna to Brooklyn, to Harvard, to NYU, to Paris, to Columbia, all the time with a giant curiosity to understand memory and the loss of memory, everything there is to know, and the biological basis of memory. That's been his life's work. That's why he won the Nobel Prize or shared it with some others. And I am pleased to have him here to talk about that on this program. Welcome.
Thank you. I feel very privileged to be here.
Thank you. Let me go back to Vienna, first of all. I mean, you born in Freud's town.
But indeed.
Yeah. And studied history. Or
I majored in an area called history and literature.
yeah.
And I was very much interested in European intellectual history. I was interested in the kinds of issues that had concerned me. And I wrote my dissertation on the attitude toward National Socialism of 3 German writers, Carossa, Zuckme, and Junge, fairly obscure people to the American public. But they were interesting because they had different positions vis-a-vis the Nazis. So Zuckme was a very progressive dramatist who actually was in Vienna. He was a German dramatist who had written some satirical dramas about Hitler, escaped from Germany, went to Austria, and then when Austria was invaded, he came to the United States. Carossa was a physician who took an interesting position, what he called a inner immigration. That is, he stayed in Germany, but philosophically he was opposed to the regime. And Junge was really a proto-Nazi. He had been in the First World War. He loved the idea of battle. He thought this brought out the best in men. And he was really one of the intellectual
Athletes came out of Erasmus High School.
That's right. Sid Luckman came from
Yes, exactly right. And others. Some great basketball players, too. Why Harvard, and then why NYU?
That's really extraordinarily interesting. So I thought I would go to Brooklyn College from Erasmus, which is where most of the academically oriented kids went to. And I had applied to Brooklyn College, and John Campana, one of my history teachers, approached me and said, "Have you ever thought of applying to Harvard?" And I said, "Not seriously." He said, "Well, why don't you think about it?" So I discussed it with my parents, and they thought, "Gee, you know, you're almost certainly going to get accepted at Brooklyn College. It would cost additional funds to apply to Harvard, to fill out the forms," and they discouraged me from doing it. So I told Mr. Campana that my parents didn't think this was a good idea, so he put out the money for me to apply to Harvard.
Wow.
And Ronald Berman and I were the 2 people from Erasmus, the class of 1400, accepted at Harvard and on a scholarship. I mean, that's fair Harvard. And she was told that one needed to get an affidavit. Somebody had to guarantee your support once you came to the United States. So she wrote to her brother and got from him an affidavit, and by the time that arrived, Hitler had already taken over Austria. A quota was established. There was an American quota, which was now very rapidly filling up, and since my parents came from Poland, it took a year before our number came up. And even as it came up, we had to leave in stages. So my grandparents left first. They left in February. My brother Louis and I left in April, and my parents didn't leave until September. My parents left a few days before the war broke out.
Reunited in Brooklyn?
In Brooklyn, yes.
Then Harvard College?
Later at Harvard College,
Yeah, and then went on to Harvard College later?
Yes. When I first came here, I went to elementary school. I first went to PS 217, and then with some tutelage from my grandfather in Hebrew, I transferred to a very good Hebrew school, a Jewish parochial school called the Yeshiva Flatbush. And I went there for 4 years until I graduated, went to high school at Erasmus, and from Erasmus I went to Harvard College.
Yeah. Some great